Hello students. Yesterday we were talking about proteins. As I told you that proteins are made up of amino acids. Amino acids. A large number of amino acids, this amino acid, this amino acid, this amino acid, this amino acid. They get bonded with each other by means of a bond called peptide. Peptide bond. It is called polypeptide or protein. Now, few examples of proteins that we want to tell you. That keratin. One question you are going to get in the examination. Many a time this question has been set. In 2015, this question was set in the examination. Keratin is a protein. It is present in hair and in nails. Then myosin and actin. They are present in muscles. Then hemoglobin. It is present in red blood cells. Then collagen is present in the human or animal world. Maximum amount of protein in the animal world. Animal world. That is maximum. Then very important protein, that is your casein. Casein. It is present in milk. Remember, very important question for you, that casein is a phosphoprotein. Phosphoprotein. As I told you that proteins are made up of carbon, hydrogen, oxygen, and nitrogen. But in certain proteins, phosphorus is also present. And that is the example. Now, a little bit of the hemoglobin, we must know it about the hemoglobin. Hemoglobin, very important. Hemoglobin. It is made up of heme plus globin. This hemoglobin is a quaternary protein. Why we call it quaternary? Because it is made up of 4 polypeptide chains. Alpha 1, alpha 2, beta 1, beta 2. All these 4 chains, they collectively are made up of 574 amino acids. Very important question. Question was in 2016. Alpha 1 and alpha 2 they are made up of 141 amino acids each. And beta 1 and beta 2, they are made up of 146 and 146 amino acids each. If we add all the total number of amino acids in one molecule of hemoglobin is 574 amino acids. Very, very important question. It is not at all given in your NCERT. Now, one more thing about the hemoglobin, that one molecule of hemoglobin, one molecule of hemoglobin can carry 4 molecules of oxygen. Remember, please. 4 molecules of oxygen. Now we come to one more protein. I am not leaving anything. Rather, I am a little bit fast because we are in a crash course. That is insulin. Insulin is a protein which is produced by the beta cells of islets of Langerhans in the pancreas. And one molecule of insulin contains 51 amino acids. Remember. Now I come to a very, very important question. Last year only it was in the examination and almost every year. We come to enzymes. The term enzyme was given by William Kuhne. Then Buchner is the name of the scientist. He isolated an enzyme called zymase from the yeast cells, from yeast. This enzyme is used in fermentation processes. Very, very fermentation. And moreover, there is one more scientist called Sumner. He discovered the enzyme urease, and he gave a statement, very popular statement, that all enzymes are made up of proteins, but all proteins do not form enzymes. All enzymes are made up of proteins, but all proteins do not form enzymes. But now, this statement is not much valid because there is one enzyme which has been produced from RNA, and that is called the RNA enzyme, and that enzyme is the ribosome. Very, very important question. Very, 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 very important question. Ribosome is a RNA protein. It is not formed from the amino acids or proteins. So what we say, that this statement has been changed, that all enzymes are proteins, but all proteins do not form enzymes. This is true that all proteins do not form enzymes, but now it has been discovered that all enzymes are not made up of proteins. Got it? Now, one more very, very important question. Please write the message if you are not understanding. Ask anything, whatever is in your mind. Or put it on my WhatsApp. I will give you the answer. Now, these enzymes are of two types. Understand it carefully, you are going to get a question out of it. One are called the simple enzymes, and second are called conjugated enzymes. Simple enzymes are exclusively made up of amino acids. Simple enzymes are those enzymes which are made up of amino acids, which are made up of proteins only. Proteins. They contain only the protein part, and that protein part is known as apoenzyme. It means we can write it that simple enzymes are apoenzymes. In the conjugated enzymes, a enzyme that has protein part as well as non-protein part. Protein part as well as the non-protein part. The non-protein part is called cofactor. Please write it down. The protein part is known as cofactor. The cofactors can be organic or inorganic. When it is the organic part, it is called the non-protein part and organic part. They are called coenzymes. When it is the inorganic part, it is called the cofactor. No doubt this is non-protein part, and definitely protein part is known as the apoenzyme. These organic parts or the coenzymes, further you can say that the further this organic part of the enzyme, that is called apoenzyme, and this is called the cofactor. There are so many cofactors. Let me write it. Let us say molybdenum. It is present in the enzyme nitrogenase. Nickel. It is present in the enzyme urease. So these are the two most important examples for the enzymes, that is urease and nickel. Nickel is present in urease enzyme. Now we come to one more equation, which is called Km is equal to half Vmax. And second, what is turnover number? Now understand turnover number. This Km is called the Michaelis constant. This represents, this is very important. Michaelis was the name of the scientist who discovered this Km. Km represents the substrate concentration at which the velocity at which the represents the substrate concentration when the velocity is half of the maximum, or you can say that half of the maximum velocity is represented by Km value. This Km value gives the affinity of the enzyme. Please, affinity. Higher is the Km, lower is the affinity. Affinity means chahat, association. Higher is the Km, lower is the affinity. Lower is the Km, higher is the affinity. Or just to remember, we can say that Km is inversely proportional to the affinity for the enzyme of that substrate. Km is inversely proportional to affinity. Now, at the same time, remember turnover number. It gives us efficiency of the enzyme. Don't confuse with the words affinity and efficiency. How can we define the turnover number? How many molecules of substrate can be broken down by one molecule of enzyme in 1 minute? एक एंजाइम दा एक मॉलिक्यूल किन्ने सबस्ट्रेट दे मॉलिक्यूल्स नु बीट कर सकदा है, ब्रेक कर सकदा है, that is known as the turnover number of the enzyme. And turnover number represents the efficiency of the enzyme. Now, the maximum efficiency is that of the enzyme carbonic anhydrase. Carbonic anhydrase. This enzyme is present in the red blood cells. Now, there must be some inhibitors. In your syllabus, there is one type of inhibition. Though there are 4 types of inhibition, but in your syllabus, he has mentioned only one type of inhibition, which is called competitive inhibition. In what is inhibitor? Which inhibits the activity of some other molecule. Here the inhibitor resembles thus the activity resembles the substrate. For example, succinate dehydrogenase acts on succinic acid. There is one more molecule which is called oxaloacetic acid. That also resembles the succinic acid in its structure. So instead of succinic acid, oxaloacetic acid enters the enzyme. Now this enzyme is specific for succinic acid, not for oxaloacetic acid. When it goes here, no reaction will take place. Nothing will occur. So this is the example. Succinate dehydrogenase enzyme, which acts on succinic acid. This activity is blocked by oxaloacetic acid because it is a competitor. It resembles the succinic acid in its structure, in its organic structure. I hope you have understood. Now we come to, and remember one thing, that enzymes are not used in the reaction. Enzymes are just like the middlemen. They are just like the bicholas. One reaction takes place, and when it is over, it comes for the second reaction. It picks up the second substrate molecule and acts on it. Now we come to nucleotides. One nucleotide is made up of base, let us say nitrogenous base, plus pentose sugar and plus phosphate. This phosphate, as we write it, it is phosphoric acid. We represent it like this, P, or we can write it like this, Pi. It is inorganic phosphate. All mean the same thing. A molecule of a nucleotide consists of nitrogenous base plus pentose sugar and phosphoric acid, that is H3PO4. Now these nitrogenous bases, they are of two types. Number one, they are called the purines. And second, they are called the pyrimidines. Purines are adenine, guanine. It is written as A, it is written as G. And pyrimidines are cytosine, thymine, and uracil. It is written as C, T, and U. And pentose sugars are also of two types. One is called the ribose sugar, and second is called the deoxyribose. Ribose sugar and deoxyribose sugar. Remember, nucleotides, they form nucleic acids. Nucleic acids are nothing but they are the polynucleotides. A large number of nucleotides get bonded with each other to form a nucleic acid. Then nucleic acids are of two types. One is DNA and second is RNA. In DNA, both the purines are present. Cytosine is also present. Thymine is also present, but uracil is not present. It means a molecule of DNA consists of adenine, guanine, cytosine, thymine, deoxyribose sugar, and the phosphoric acid. But in case of RNA, they are formed by the ribonucleotides. Ribose sugar is there. Remember one thing, ATP also contains ribose sugar. That is also a ribonucleotide. You can write it. ATP, which is called the energy currency of the cell. It is a ribonucleotide. In RNA, adenine is present, guanine is present, cytosine is present, but thymine is absent and uracil is present in RNA. And instead of deoxyribose sugar, ribose sugar is present. I just write it again. This is DNA is equal to adenine plus guanine plus cytosine plus thymine. These are the bases. Plus deoxyribose sugar. And then phosphoric acid or P. One question is lying over here, that's why I'm spending time. Adenine and guanine, they are purines. And cytosine and thymine, they are pyrimidines. Now coming to RNA. RNA is equal to adenine plus guanine plus cytosine plus uracil, not thymine, plus ribose sugar and plus phosphoric acid. Again, these are the purines and these two are the pyrimidines. I hope you have understood it. Now we come to in brief about the structure of the DNA. I have told you the composition of DNA, what is present in DNA. DNA is made up of deoxyribonucleotide units. In other words, what we can say that DNA is a poly-deoxyribonucleotide. Poly, बहुत सारी जुड़ते हैं. The structure of the DNA was given by Watson and Crick in 1953. And both of these, they got the Nobel Prize in 1962. A molecule of DNA is a double helical structure with 5' and 3' and this 3' and this is 5'. Yes. What do we mean by 5' and 3'? It means its molecule, last molecule, is connected to the carbon atom at 3 position of the deoxyribose. And here it is at the 5 position. Now the length of this DNA molecule is 34 angstrom units. Nowadays, it comes in the form of nanometer. It is 0.34 nanometer. And its width is 20 angstrom units or 0.2 nanometer. There are 10 bases present in one helix. 1, 2, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Because the total length is 34, there are 10 base pairs. Therefore, the width, the distance between two consecutive pairs, any consecutive pair, it is 3.4 angstrom units. So this is 20 angstrom units, and this is 34 angstrom units. And one more thing I would like to tell you, there are 5 types of DNA: zDNA, aDNA, bDNA, cDNA, dDNA. This structure is of the bDNA, which was proposed by Watson and Crick. In zDNA, there are 12 base pairs instead of 10. It is 11 base pairs. It is 10 base pairs. It is 9 base pairs. And it is 8 base pairs. Very simple to remember. 12, 11, 10, 9, 8. Among these, all DNAs are right-handed. Right-handed structure. And this zDNA, which is called the zigzag DNA, and it has got the left-handed structure. This is all about the DNA. Therefore, the difference between DNA and RNA is that DNA contains thymine, RNA contains uracil. DNA is made up of poly-deoxyribonucleotide units. It is a poly-deoxyribonucleotide, and RNA is a ribonucleotide. And next, DNA contains deoxyribo sugar, and RNA contains ribose sugar. So this is all about this chapter. Now we come to, Madam has already taught you about the transport in plants. Now we come to the next chapter of plant physiology, that is mineral nutrition. Mineral nutrition. There are certain minerals which are essential for the growth of a plant. And there are PET rules, PET criteria to determine the essentiality of the minerals needed for the growth of the plant. And this classification or this criteria for the essentiality of the elements, that was given by Arnon and Stunt. Please note it down, it is not given in your NCERT. This question was definitely asked in 2001, 2001-5. Arnon and Stunt. Without that element which is deficient, growth of the plant just write your message again, please flash it. Flash it your message, Muskan, or what is his name. Please flash your message. I have not read that message. Flash it again. Arnon and Stubbs. The element which is deficient in quantity, the growth of the plant is inhibited. And even the life cycle of that plant cannot be completed without that particular element. These nutrients or mineral nutrition, they are of two types. Number one is micronutrients. And second is macronutrients. There are total 17 minerals which are responsible for the growth of a plant. 17. If in the examination it is written 16 and 17 choice is not given, then 16 is also the right answer. Because late nickel has been added. Earlier it was 7. Nickel is the latest element that has been added or that has been considered to be a nutrient. There are 9 micronutrients which are needed in small quantity. And there are 8 macronutrients. Just write it, please. The micronutrients, it is very easy to remember. I am giving you a formula to remember this. This is SPONK. Then more thin elements are potassium, calcium, and magnesium. Potassium, calcium, and magnesium. PCM. S means sulfur. P means phosphorus. O means oxygen. N means nitrogen. C means carbon. H means hydrogen. P means potassium. Let us take K, not K. P, let us take K. Potassium. Then C, it is calcium. Let us write this Ca. Then it comes the magnesium. So these are the 9 elements. Again, I just tell you 9 elements which are considered to be micronutrients. SPONK, it is easy to remember. These are the 6 major elements. Then comes K. K means potassium. Ca means calcium. M means magnesium. These are the 9. All other 8 which are left behind, let us write it. Copper, molybdenum, then comes your zinc, boron, very important. Then comes manganese, then comes nickel, copper, molybdenum, zinc, boron, manganese, and then comes the chlorine, iron, and iron. Or I just tell you the simplest way to remember. Remember these. The question in the examination comes, which of the following is a micronutrient? Or which of the following is not a micronutrient? Or which of the following is a macronutrient? And which of the following is not a macronutrient? One question per count. And now, if you remember this, all others are macronutrients. You will not write anything else. Remember these: SPONK, sulfur, phosphorus, oxygen, nitrogen, carbon, hydrogen, then potassium, then comes calcium, then becomes the magnesium. Now let us just discuss the role of one by one important ones. Coming to carbon, hydrogen, and oxygen. They are called the framework elements. Framework elements. Then comes sulfur. Sulfur is present in two amino acids. Yesterday I told you, cysteine and methionine. Then comes potassium. I am writing the important ones only. Potassium. It is responsible for opening and closing of stomata. Then comes phosphorus. Now it is responsible for premature leaf fall. Very, very important question. It is responsible for premature leaf fall. Now we come to molybdenum. This is carbon, hydrogen, oxygen. They are called the framework elements. Sulfur is present in cysteine and methionine. Potassium is opening and closing of the stomata, and phosphorus is responsible for premature leaf fall. Then comes molybdenum. It is responsible for nitrogen fixation. Then zinc. Yesterday I told you IAA, indole acetic acid, is formed from tryptophan in the presence of zinc. Then comes manganese. The higher amount of manganese that causes toxicity. जे ਆਪਾਂ भी ज़्यादा रोटी खा लिए तभी अपन भी toxicity. जे manganese ਦਾ level बहुत ज़्यादा बढ़ा दिए plant के बीच, then toxicity, because it will inhibit the uptake of calcium, which is very important. I've already told you that nickel, it is present in the enzyme urease. So these are some of the most important elements which you have to remember. I just repeated. Carbon, hydrogen, oxygen, they are called the framework elements. And nickel is urease, sulfur, cysteine, methionine, potassium, opening and closing of the stomata, phosphorus, premature leaf fall, manganese causes toxicity, zinc forms indole acetic acid from tryptophan, then molybdenum, it is responsible for nitrogen fixation. Now the most important question for you at the last. Manganese, calcium, and choline, they are responsible for photolysis of what? Photolysis of what? During photosynthesis. I can say the most important question for you. This is the most important question for you. And this is the most important question for you. And this is the important question. These are some of the important questions which you are to remember. That manganese, calcium, and choline, they are responsible for the photolysis of what? Photolysis. Pani da tut jana in the presence of light. We will be studying in photosynthesis. Then zinc, then molybdenum, then nickel, then sulfur, then potassium is for the opening and closing of the stomata, and phosphorus, that is the premature leaf form. I hope you have understood. If you have got any doubt, just put the message, please. Then I start the next topic. You can easily and comfortably put the message. Thank you very much. Thank you. Thank you. So nice of you. Thank you. Thank you. Thank you. And now we come to the one more term in this chapter. अगर तुसी साडा हौसला एवें बढ़ाउंदे रहोगे तां असीं होर भी ज़्यादा काम करदे रहांगे, because you have come in contact with me for the first time. I have got 30 years of my experience. And my son is also in medical college. He's doing MBBS. He was a student of Dr. Professor Adarsh Bhatti also for chemistry. So I hope and wish that you all reach to medical college. You try your level best. One should not leave trying. It is not written on anybody's forehead that this thing will happen. Sade hath de vich sirf hard work hai. We can do hard work. And I'm a robust believer that hard work never goes wasted for anything. And now I explained the term what is hydroponics. It is a soilless culture. Soilless culture. It means culturing of the plants without soil. And here a chelating agent, very important question, not written in your NCERT, EDTA is used. Now EDTA stands yes, ethylene diamine tetraacetic acid. And one more important question which is not in any NCERT, that you will get 15 to 20 questions as such which are not in your NCERT. Please remember. This method of soil culture was proposed by Julius Sachs. Remember, Julius Sachs. So dear, this is all about your mineral nutrition, which you are to do in small and in a nutshell. Now we come to the next chapter. Definitely you are going to get 3 questions out of it, the chapter I am going to teach now. That is for the respiration. Any problem you get, you just put it on my WhatsApp. I'll be rather happy at any time. At night, in the morning, in the afternoon, anytime. Just you put the questions and your problems. Don't ask me. Don't ask me, how are you? I'm always fine. Students always wish good for me. My name is Dr. A.C. Goyal. So you can take my number from the Gurukul and you can always put the questions on this Studyrocks or contact Raj Madam or Professor Adarsh Patti. I'll be rather happy to answer your questions anytime. Now coming to respiration. Now what is the difference between breathing and respiration? Breathing is a mechanical process. It uses energy. It is breathing. Respiration, it releases energy. And you know that energy is always produced in the form of ATP. We are a bit fast because it is a crash course. ATP, it is adenosine triphosphate. I have already given you one more important question. That ATP is a ribonucleotide. In this case, ribose sugar is present. It is a ribonucleotide. I have already told you. And moreover, the energy is stored in the body in the form of ATP. Energy in the body is stored. Rather stored, it is in the form of ATP. Energy is produced rather, let me not stored. Energy is produced in the body in the form of ATP. ATP is a ribonucleotide. And ATP, very important question. It is also called energy currency of the cell. Energy currency of the cell. Now this respiration is completed in 3 steps. Number 1 is glycolysis. Remember, there are 6 questions in glycolysis, nothing to read more than that. 6 questions, only 6. 6. What? It is also called EMP pathway because it was proposed by 3 scientists. Amton, Meyerhoff, and Parnas. These are the 3 scientists which are in your first question. Number 2 question. This is 1 question. Number 2 question. Respiration is always an oxidative process. What is oxidized over here? It is the oxidation of glucose. Glucose is the simplest carbohydrate. We eat anything, ultimately it gets converted to glucose. We go to the hospital, आप अस्पताल जाने या कोई प्रॉब्लम होती है जी, तो डॉक्टर अपन को ग्लूकोज की ड्रिप देता है। ये तो नहीं कि अपन कोई संतरे दे जूस की ड्रिप दे दे, या अपन को घिओ की ड्रिप दे दे, या अपन को कोई और ड्रिप दे दे, no. Because glucose is the instant source of energy in the body. Immediately it gives energy. Immediately it enters the glycolysis. Oxidation of glucose. Third, it is anaerobic process. Please don't confuse with anaerobic respiration and aerobic respiration. Glycolysis may hona hi hona hai, whether it is the aerobic process or anaerobic process. But in glycolysis, no oxygen molecules are used. It's a very important question, very important question for your NEET examination. How many number of oxygen molecules are utilized in glycolysis? You will say 0. No oxygen molecules are utilized. It is an anaerobic process. Anaerobic oxidation of glucose. One molecule of glucose. Remember, it is one molecule. One molecule. Now next, fourth. To form what? Oxidation of glucose will lead to what? To form pyruvic acid, which is a 3-carbon compound, and glucose is a 6-carbon compound. It means definitely 2 molecules of pyruvic acid will be formed. And fifth point, this glycolysis always occurs in the cytoplasm of the cell. Glycolysis always occurs in the cytoplasm of the cell. Cytoplasm. And last point, but not the least, that glycolysis is the common pathway. You can write it by yourself here. 6th point, common pathway for aerobic and anaerobic respiration. After the examination, if any question from glycolysis comes except these 6, you can get angry with me. You just put your question on my WhatsApp. Number 1, it was discovered by 3 scientists. Number 2, it is the oxidation of glucose. Glucose is a 6-carbon compound. Only 1 molecule of glucose enters at one time. It is an anaerobic process. No oxygen molecules are utilized. And similarly, no carbon dioxide molecules are produced in glycolysis. Then it forms 2 molecules of pyruvic acid. Pyruvic acid is a 3-carbon compound. And then the last, that it occurs in the cytoplasm of the cell. And it is the common pathway for aerobic respiration as well as anaerobic respiration. I hope you have understood. Give me the message.